evening, welcome to the class. Hi everyone, I hope that you can clearly see me and can hear me. Good evening Dipanshu, good evening Purna, good evening Sneha. Good evening, how are you all? Good evening Aditi, I'm good, how are you? Hi Rashmi, good evening. Hi Tripti, thank you for telling me Tripti. Renuka, we don't have Menti session today, but it's a one shot. Prachi, hi, good evening. Good evening Tripti. Nice. I hope that all of you are fine, right? And just 1 minute. I hope that you can clearly see me and can hear me. Hi Ashraf. Nice Aditi, thank you so much. Hi Priya, good evening. Preeti, Narendra, Advay, Jaiprakash. The student show, nice. Hello, good evening. Hi Anand, ciao. Welcome everyone. Yes, yesterday we didn't finish that, right? But today we are just having one shot. So the basically read line by line with the NCERT will not be doing in today's class, right? So of course, as you can see from the topic, we will be having a whole session, one shot. Okay, Rashmi, so today it's just one short video for all of us. I know 3 or 4. Yes, Purna. No, we haven't done this one shot. As of now, I think we didn't have any one shot. Okay. Without taking any of your time, everyone, let's get started. I want all of you to focus over here. Hi, Ashwarya. Yes. So I want all of you to fully focus over here. It will not take much of your time. Right, and we will be focusing to finish this chapter, right? So, all focus. Now, before we start, of course, you saw over there, right, in the video, and of course, that's all about the ANTHE. Now, we are talking about this interesting and super amazing exam, which is Aakash National Talent Hunt exam. Now, this exam will give you a chance to go to the NASA, right? And of course, that's a really very interesting thing. Now, apart from that, of course, there are a lot of other benefits. We have cash awards. You'll be getting all India ranking and up to 100% scholarship. And of course, the online exam is there, right? An offline exam is also there. Please do participate. Apart from that, now we are targeting our midterms, right? And of course, we have all of these classes. Now, how many of you have joined the Telegram group? Yes. How many of you have joined the Telegram group? Now, once. Good evening, good evening. Good, very good Purna. We all should participate, right? Yes. Hi Husna, hi Shivansh, hi Ruby Jem, hi Dipanshu, Gautam, Parth. Awesome. I can see a lot of you have joined. So we are providing the chapter revision and PDFs also. So we have all of the tissues ka PDF. As of now, all the tissues PDF are all there on the group. So please, now you can download those PDF. We'll be focusing on the important concepts, important questions that are really very important from the exam point of view. Then of course we have doubt sessions, one short marathon like this, and of course we have mock test. So definitely just subscribe to the channel, and very importantly this. For every week what will happen, we have first 1,000 subscriber, right, who can actually avail the mini learning program by using the code YT first. So you'll be able to have those amazing classes. You can take a look at the classes that we have, and of course, it'll be nice fun. And I already talked about this. Join the group and let's get started. Mimor ka adda, wish I can tell you, this is not a place. And I think that you know that we should not be asking about the salary and such public platform. Not a good thing. Let's not do that. Great, everyone. So are we clear? Okay, give me a quick thumbs up, everyone, and then we'll start. 5, 4. I'll just need, I think, approximately 45 minutes of yours. Yes? Great. Oh, very quickly a thumbs up, everyone, and then we will start, right? I can see Muskan, no issues. Mimar Skarda, it's fine. Yes, Aditi, Simran, Prachi, Anant, it's okay. Let's not, of course, he must have asked, out of curiosity. Let's not do anything, right? And he said, sorry, that's perfectly fine. Yes, Sharanjit, Ajit, Rashmi, Dipanshu, Gautam, Vivek. Very good, Monalisha. Very good. Let's get started, everyone. So that's a very interesting chapter that we have today. Devdas, you're right on time, right? Tarun, Pokemon. Nice. Okay, Gigi, Ajay, Chandan, Sneha. We don't have any homework question today. Chandan, we have already started the tissues chapter, right? We have finished plant tissues and we have started with the animal tissue. Epithelial tissue is done, and next week we will be covering up the other tissue. So yeah, stay tuned for that. Muskan, tomorrow we'll have the life process one shot, right? Great. Okay, everyone, on that note, just need your time. I want fully focus of you, and let's get started. So when we talk about the fundamental unit of life, it's a very interesting chapter talking about the cells, right? Now, of course, one of the important things that we will be asking in the questions, and of course in n number of examination, is that who discovered the cell? And we have the answer. Robert Brown is the one who actually discovered the cell in the year 1665, right? He was the first one to observe the dead cells, right? He observed the dead cells. Now, and he was the one who coined the term cell due to its appearance as of a prison, like small, right? So that's why we got the name cell. And he observed the dead cells. Whereas on the other side, we have Leeuwenhoek, who actually discovered, well, not say discovered, who actually observed the first living cell, right? And of course, he used the improved microscope. So that's one thing we all are clear. Leeuwenhoek, we know that, observed the first living cell. Yes, it's Robert Hooke. It's not Robert Brown. Yeah, Robert Hooke. Yes, आज मेंटी नहीं होगा. Moving ahead, we have the discovery, right? Just give me 1 minute. So we have the discovery. Discovery of the cells, of course, we have this. Apart from that, we have we call it as a cells, as a structural and the functional unit of life. And they are really very important. We have animal cell, and over here we have the plant cell. Then let's talk about the types of cells that we have. Now, of course, we have, in numbers, we have unicellular organism, like amoeba and bacteria, which is one single cell that are capable of performing all the function by themselves. And then, of course, we have the multicellular organisms, right? Like us, we have n number of cells, and of course, each of the cells perform a specific functions. So, of course, the number of cells will be help us to identify the type of organism, unicellular organism and the multicellular organism. Then, of course, we have different cells and they have their specific functions. Like nerve cell will help in the nerve conduction. RBCs will actually help in the transportation of oxygen, right? And of course, we'll be carrying back the carbon dioxide. And then we have the muscle cells, and these are the smooth muscle cells we're talking about. They actually help in the involuntary movement. Talking about over here about the cell membrane. Yes, I hope that all of you are clear. Yes, deoxyribonucleic acid. I think someone was asking for the full form of DNA. Which is the plant cell and which is the animal cell? Can you spot the difference? Yes, we can easily. Now, once we'll be looking into it, you'll be able to find. Nice. I hope that it's clear. Good evening, Tejaswini. Yes, very good. I can see thumbs up. Clear. Function of RBC, Sneha, they actually help in the transportation of nutrient and the gases. Right. He discovered the Vani, right? So, of course, I think your friend have mentioned over there. It was in some 74. Right, you can check in the book. They have mentioned about that in which year Leeuwenhoek actually saw the first living cell. Plasmolysis. Yes, we'll be discussing about that. Okay. We'll discuss about that once we're discussing about the cell wall. Let's talk about the cell membrane, and we are focusing on the animal cell. So those of you, please pay focus now, please pay attention, and this is the animal cell that we are discussing about. So here we have the animal cell, and an animal cell, the outer covering is called as the cell membrane. Very easy, right? We know that it's very easy. Now, of course, it's a protective covering that actually holds all the content of the cell together. Now, we know that all of it will be present inside the cell, and of course, a plasma membrane acts as a barrier. Then, apart from that, it actually helps in the transportation of the selected molecules only between the surrounding, and that's why we call them as the selectively permeable, right? So, plasma membrane is a selectively permeable membrane that allows the entry and the exit of only few particular molecules. Cell membrane is also called as plasma membrane. Yes, Talib. Okay. Anant, cell membrane is also present in the plant cells also. Very good. Anant, I have answered your question. Tarun, can we focus in class? Yes, cell theory was given by 3 scientists, Sharanjit, right? 2 specifically which started with it. We have Skelton and Schwann, right? And then of course Rudolf added a very important postulate to it. Very good. Yes, Sneha have answered that question. Sneha have answered that question of RBC. They actually help in the transportation of nutrient and gases. We don't have NCRT today, but we will be having it really very soon in the next week. So focus on that. Permeable means, Tia, that it is something that can allow the movement. Right? Awesome. Maralisha, I think I have answered your question. Very good. So we have the cell membrane. Are we clear? Very good, very good. Yes. Protoplasm is nothing but the living content of the cell that includes the nucleus and the cytoplasm, basically the all important organelles. Nucleus is there. Very good, Rashmi. It is made up of the lipids and the protein. So over here when we talk about the plasma membrane, over here can you see we can see the structure. So of course it has the combination of lipids and the protein, and of course it has 2 layers of that, and of course it has the different types of transport. Now there are 2 types of transport that occurs, active transport and the passive transport. In the active transport, there is a utilization of the energy, right? And the molecules will move from the lower concentration to the higher concentration. That's why they need the energy. The other type of the transportation is the passive transportation. Now, no energy is required and it occurs really very smoothly from the higher concentration to the lower concentration. So, are we clear, everyone? Yes? I will be coming up to the different points. Dipanshu, right? Isha, just give me a minute. Right? Are we clear? The 2 types of transport are there, active and the, we have 2 types of transport, active and the passive transport. Now, when we talk about the passive transport, we have 2 important things over here. Now, many of you are asking about this. Here we go, we have the passive transport. We have 2 important parts. We have osmosis and diffusion. Right? Now, in osmosis, we know that the movement of the solvent molecules from the higher concentration to the lower concentration. And when we talk about the osmosis, we talk about the water per se. Then it is always takes place with the help of a semi-permeable membrane, and of course osmosis occurs only in water in the aqueous solutions, or basically in the water content. Whereas diffusion, we will see both the solute and the solvent movement from the higher concentration to the lower concentration. There is no requirement of the membrane, and of course it happens in the liquid and the gases. Are we clear? Yes? So, if I quickly wrap it up, we have active transport. Right? In active transport, we will see the movement happening from the low concentration to the high concentration with the help of ATP. Energy is required in this. Right? I'm not sure what is this exam you're talking about. Can we focus over here? Manya, ATP is used as an energy molecule, energy currency of the cells. So of course, it is used to perform various metabolic activities. It's the energy that is required. Very good. Yes, it is good. ATP, adenosine triphosphate. Thank you, Rafiq. Yes. Sharanjit, you need to wait, bache. We will be coming to lysosomes, but just need to wait for some time. It's okay, Himani, no issues, have just started. How does plant cell divide? We will be talking about that, Mona Lisa. Just give me a minute, right? Yes, Bhuvnesh, we'll be discussing about that also. Now, everyone, see, after each and every topic, we'll give you time to ask doubts, right? So, I want all of you to focus over here. Are we clear with this? Are we clear with what is cell, about the cell theory, right? Now, about the shape and size of the cells, different types of cells, unicellular organism or multicellular organism, and about the plasma membrane, right? And the type of transport. Now comes a very, very, very important part, which is the different types of solutions or the tonicity of the solutions, right? So what we have over here, everyone, focus. We have different types of solution. Now first, we're talking about the hypertonic solution. What happened in hypertonic solution? Solutions with the higher concentration of the solute. So over here, if you can see in a water jar, right? Let's suppose we have the mango, right? And we have added a great amount of salt into it, and we have dissolved the salt. Now, of course, the salt over here is the solute, right? Now we have added into the water, which is the solvent. Now, what has happened, right? What will happen eventually? The mango will lose its water and it will shrink because outside there's less water. So, of course, it will lose the water, and in that case, the mango or the cell that is present in the hypertonic solution will shrink. Easy. What will happen? We'll see the shrinking. Yes? Okay. Got it? Bhumi? Should I explain it again? So, very simple. We have this jar, right? We have this jar and we have this cell. Now, of course, it has salt into it and it has less salt, very less salt. Let's just suppose 1 or 2. Now, it has more of water and less water is there outside. What will happen? The water will move outside and the cell will shrink. And that is the hypertonic solution. Are we clear? Yes. Are we clear? Very good, very good. That is the hypertonic solution. Now, can we talk about the hypotonic solution? Hypotonic, what will happen everyone in hypotonic? I've told you this 1,000 times. What will happen in hypotonic solution? A cell is there, right? And it has more amount of water. Yes, what will happen? The cell will swell up, water will move inside and we will see the change in the shape of cell, right? We will see that it will swell, right? It will become like it will have more of the water. Yes. Very good. It will expand. Now, if we talk about this, that we have a cell, right? And the concentration of the water is almost equal to the concentration that we have in the cell, right? A kind of a same. What will happen? Nothing will happen. Equal movement will be there. So, of course, that type of solution is called as isotonic solution, right? Right, everyone? This is we have already studied. So, over here we have for the hypotonic and over here we have it for the isotonic. Very good. Isotonic, yes. Isha, equilibrium means that it will be in a balance, like both the things will be equal. Right, so we have 3 types of solution, hypotonic, hypotonic and isotonic, right? Now I'll take some doubts. I will be taking some doubts. Yes, it's easy. If you have any doubts, you can write over here. Concentration means that, of course, it will be in terms of the concentration. For example, if you talk in the easiest way for you to understand, let's suppose you have more amount of the apples and less amount of the banana at home. Like if you talk in terms of the apple and banana, you have more of that and more of this concentration, right? Okay. Yes. Are we clear, everyone? Okay, you're asking about the... Yes, I will explain that again. I will explain that. Yes. Okay, I can see some few questions. I'll just take them up. Okay, first I think I can see is the tonicity. Right, this is what we have. Now, of course, it is nothing but the capability of a solution. For example, if we have a solution over here, the capability of a solution to modify, right? The capability of a solution to modify or to change the cell shape, basically, right? To change the alteration in the cell volume per se. Right, so for example, this has to do with the solution. When we're talking about it, it has to do something with the solution. When we add something to the water and to the solution, that will be affecting the cell volume. So it is the tonicity. Very good. Now I hope this is clear. Yes. Right, I hope this is clear to you. Now, I think there are few of you who have some doubts. The cell was discovered by the Robert Hooke. Right? Very good. We will be discussing about that. Navneet, we will be discussing that in tissues, bache. Is it the concentration of a solution? Yes, it is. Ruby will be discussing about that when we'll talk about the Golgi Apparatus. Hypertonic solution. In hypertonic solution, what will happen? The cell will get the water from the beaker, right? And of course, it will swell. Sorry, it will swell. So what will happen in the hypertonic solution? The cell will swell. So it will take water. Then why we put mangoes in the vessels with the water? So of course, mango over here is just an example, right? We can add any plant cells or any animal cells, right? So basically, we are doing it to actually check the type of the solution, right? And of course, if we add something to it, what will be the effect on the cells? Yes. Mona Lisa will be discussing about that when we'll talk about cell division. Very good. So everyone, are we clear up to here? Now I can see many of you are asking a lot of random doubts which are not per se we have covered as of now. Now I want you to ask the doubts of the topic that we have just discussed. Plasma membrane, osmosis, diffusion. So Sneha, in hypertonic, remember it will shrink. Hypo, remember hypo with the hippo. Hippopotamus, right? I'm sure you have heard about this animal. Yes? Yes, Mania, we will have the doubt session class also. Yes, so we have the Sneha, so what you can do, you can remember this. Why we put RBCs in the concentrated solutions? Yeah, what will happen if we put the RBCs in the concentrated solution? Of course, if we have more salt and more of it, we will see there will be the busting of the cell membrane. It will not happen in the plant cells, but it will happen in the animal cells. Yes, Shivanshu, I think I have answered your question. Very good. Welcome Bhumi to the class. If you can write your question, I'll be able to help. Difference between plant cell and animal cell we'll discuss. Diffusion may what will happen, we will see the movement happening from the higher concentration to the lower concentration. So if we have a jar, right, and of course when we add, if you drop a ink into the water, right, slowly what will happen? We have in the dropper, we have the higher concentration and it will fall and it will move in all the direction. From higher concentration to the lower concentration. Okay. Yes, I can see your chat and I hope that this is clear now. Yes, very good. Hypertonic solution I have explained, the cell will swell. Nice. Now let's talk about another important topic, that is nucleus, and now I want all of you to focus over here on the nucleus, right? Now we'll be discussing about the important points of nucleus, and Sharatjeet, you will be able to find the answer about the question that you have been asking. So when we talk about the nucleus, is the master of the cell, we call it as that, right? And it has nuclear membrane which will be covering it, right? And inside it where we have the genetic material, we have the nucleolus at the center, and of course we have the nucleoplasm. Let's quickly discuss about all the important parts. So we have nuclear membrane, which is a double membrane, right? Covering outside the nucleus, it has small pores which are called as a nuclear pore, through which we will see the material will be moving inside and out, right? It will allow the movement of materials between the cytoplasm and the nucleus per se. Then of course we have the nucleolus. The next part is the nucleolus. The nucleolus has the site of protein synthesis, right? Basically, it helps in the synthesis of the ribosomes, and ribosomes comes out and they are present on the ER and in the different parts of the cytoplasm. On ER, they help in the synthesis of proteins. So ribosomes plays a very important role. Then they occupy 25% of the nucleus, and of course they are called as a brain of the nucleus. So they are again very important, right? They are called as a brain of the nucleus. Then we have the nucleoplasm, which is nothing but the fluid which is there, and inside this fluid we'll have the chromosome. So it's a fluid which is filled inside the nucleus, contains the genetic material or the hereditary material, and it is quite similar to the cytoplasm of the cell. Right? Now we know that why we call nucleus as the brain of the cell, because it controls various activity. Now we can say that it controls all the activity of the cell. Whatever is happening, whatever be the happening of the cell's function, it is controlled by the nucleus. That's why we call it as the center or the brain of the cell. Right? Are we clear? Tia, heredity means genetic material. Yes. So part double membrane organelles, we call them, like we have different organelles which have double membrane, right? So we have this one, nucleus. Tia, pores means small hole. So for example, this is a pore, right, on the membrane. Yes. Where is ribosome synthesis? In nucleus, in the nucleolus. Very good, very good. Nice. So, as of now, we have talked about the nucleus, right? Focus everyone over here rather than on the chat. Now, we know that nucleus has a nuclear membrane that actually covers the genetic material. Inside that, we have the genetic material. Inside nucleus, we have the nucleolus, which is a site for the basically site of the ribosome synthesis. Then we have the nucleoplasm, which have the genetic material. Now, when we talk about the nucleoplasm, right, we will be able to find the chromosomes there or the genetic material there. Now, chromosomes are very beautiful structure, right, when they are condensed, right? They are the condensed form of the DNA. Now, DNA over here is a deoxyribonucleic acid, and it is a genetic material of us, right? It contains all the information that we have, the hair color, the skin color, etc. We have it in the DNA. Now, DNA wrapped around the proteins, right? When the DNA will be wrapping around the proteins, it will be forming a structure. And when it forms more and more condensed form, it forms the chromatin. Right? It forms the chromatin. Yes, and the chromatin will further condense and form the chromosome. So, if someone will ask you, right, the chromosomes are nothing but the genetic material which we'll be able to see only during the cell division. If cell division is not happening, they will be opened, right? They'll be scattered. They'll not be condensed in this particular form. We'll be able to see chromosomes only during the cell division. Are we clear with this? Are we clear with this, everyone, about the chromosomes? Ribonucleic acid. Sharanjeev, we call it as a ribonucleic acid. Anant, I did that nucleus, right? Control and coordination will start, Amrita, once we are done with the midterm, because in the first time we have this only, life process. Yes, the green part, centromere. Difference between Prachi, the chromatin and the chromatids. So, chromatid of course is a one arm, right, that we say. See, this is the chromatid. Okay, that is chromatid. And chromatid is something which is this one, right? It gets more and more condensed. Yes. If you open the chromosome, we'll be able to see DNA wrapped around with the proteins, and that part, that structure, the bead-like structure that you see over here, right? This is chromatin. Are we clear with this, everyone? Very good, very good. Now, of course, when we talk about the chromosome, this is an important thing you can talk about, right? Now, we have the small segments of the DNA sequence that contains the important information, and we call them as the gene. Yes, Tejaswini, just answered your question. The small segments of the DNA, right, that carries a specific function. Right, for example, your hair color or your skin color, that particular trait will be carried by the genes. Yes, Nikita, brain of the nucleus is nucleolus. Very good. Yes, Neha Rajput, hello. And now many of you are asking about this, right? Prokaryotes and the eukaryotic cells. So over here we have So prokaryotic cells are those whose genetic material is freely suspended. They are not present in the nuclear membrane. So once the genetic material is freely suspended, we call it as a nucleoid. Are we clear? Many of you was asking about this. So here we have nucleoid. A cell which has a primitive nucleus and devoid of most of the cell organelle, we call it as a prokaryotic cell. Are we clear? Right? Over here we have the example and we see this in the bacteria. Then of course we have eukaryotic organisms like ours, right? Which we have the nucleus, the genetic material is contained inside the nuclear membrane, right? And of course we have this. Yes, Devdas, you're asking about the karyon. Over here we have nucleus. Karyon means nucleus. Tejaswini, whose chromosomes are single? Now, the chromosomes number varies from individual to individual and organism to organism. Right? Amit just have mentioned over here. Swapnil, thank you for helping. Amit, prokaryotic cells over here. Now, prokaryotic cells are those which does not have the nucleus. And they have genetic material freely suspended and we call it as nucleoid. Tejaswini, very good help for helping your friends out there. Okay, are we clear? Now, you're asking about the chromosomes. Now, chromosomes are the genetic material, right? Of course, they are condensed form. Okay, can I start with that? Okay, everyone focus over here. Now, what is this? Can you tell me? Quickly, everyone tell me what is this? The one thing that I've drawn on the board, right? Yes. DNA, very good. Now, our DNA is really very long, right? It's very, very long. Now, it needs to compress and it needs to be, for example, it needs to go inside this circle, which is the nucleus. So, for this, what will happen? It will actually get packed on the proteins. So, this DNA will wrap around on the proteins. Okay? And as it is wrapped around the proteins, what will happen? It will form these structures. Right? It will form a very condensed structure, a long chain of this. And this we call it as chromatin. Are we clear? Quickly everyone, are we clear? Right, is the chromatin. Yes? Now further this chromatin during the cell division will come and it will form a beautiful structure which is called as the chromosome. Are we clear everyone? Yes? So, we have the chromosomes. And now this chromosome during the cell division we'll be able to see. Are we clear? Very good. So, DNA will get packed with the proteins. Plus DNA will form the chromatin, and once they arrange themselves, they'll be arranging in the form of chromosomes, and during cell division, we'll be able to see them. Kitchen of the cell is the chloroplast. Yes, it's correct. We call it as plastids also, right? Plastids. Chloroplast is there in the plants. Quickly, everyone, a very quick thumbs up will help me. Yes, thumbs up, everyone. Very good, very good. Yes, very good everyone. So till now we have discussed about the cell, right? The different types of cell shape. We have discussed about the discovery of the cell also. We talked about the plasma membrane and we have just spoken about the nucleus. DNA full form deoxyribonucleic acid. Very good. Yes. Very good, very good. Nice. Now let's move ahead and let's talk about the cell organelle. I can see many of you are asking about doubts about the organelles. I will be answering all of those doubts now, right? Very good. Yes, you can help your friends out there, right? Sharanji, thank you for helping. Please write the full forms of it. I have already mentioned DNA, deoxyribonucleic acid, RNA, ribonucleic acid, ATP, adenosine triphosphate. That's good. Okay, now talking about the cell organelles, we know that cell organelles are present inside the cells and of course they have a very specific function. We are starting with mitochondria. Now for this, you can quickly take a screenshot of it, right? I'll just quickly move. Now this is important, everyone. Now all of these slides have been made in such a way that you can quickly revise them in the examination. So mitochondria will act as a powerhouse of the cell and will play a very important role in the synthesis of the ATP generation. So ATP production, ATP, which is the energy currency of the cell, occurs in the mitochondria. That's a very important function of mitochondria. Mitochondria have its own DNA, right? Own DNA, and it can make its own proteins, right? Can make its proteins. Okay? It has its own DNA and it's capable of making its own protein. So ATP is adenosine triphosphate. Very good. Now let's talk about the other cell organelle. We have the smooth ER. So now over here we have ER. Smooth ER help in the synthesis of lipids. Many of you are asking, right? Sneha for you. You can stop spamming now and focus over here. So, smooth endoplasmic reticulum is where we'll see the synthesis of the lipids, right? We'll see the synthesis of lipids here and it plays a very, very important role. Now, smooth ER does not have the ribosomes on top of them. That's why their surface is really very smooth. Opposite, we have the rough endoplasmic reticulum, that is RER, where of course ribosomes are present on them and they are the site of protein synthesis. So lipids are made by the SER and rough endoplasmic reticulum makes the protein, right? Lipids and protein together will be making the plasma membrane by the process of, you can tell me, right? Membrane biogenesis, right? Very good. Yes. Are we clear? Sharanjit, why we call mitochondria? Because it's make ATP. The energy which is required by the cells to perform a lot of activities comes from the mitochondria, the ATP that it produces. Very good. Yes, Shweta, they are present in both eukaryotes as well as in the prokaryotes. Yes, Ashraf have just explained ER, two types of ER, smooth endoplasmic reticulum and rough endoplasmic reticulum. Smooth will synthesize the lipids and rough will synthesize the proteins. Very good. Thank you, Rashmi, for helping. Anant will be covering up that topic. You need to wait some time. Very good, everyone. Now moving ahead to the Golgi apparatus, right? The Golgi bodies plays a very important role in the packaging, modification and in the transportation. That's why we call it them as the postman of the cell also. So Golgi bodies will be receiving the proteins and the lipids. They will receive it, right? They will modify it, they will pack it, they will transport it to the different parts of the cell. Very good. Very good, very good. Are we clear? Mr. Golgi, yes. Anant, you can find the answer. Students, please help Anant. Who is the scientist who actually found the Golgi apparatus? Very good, Tejaswini. Camleo Golgi, right? He's a scientist who actually discovered the Golgi bodies and that's why it got its name, Golgi. Yes, so, yes. Very good. So Shivansh, I'll help you with this. One side is where it receives. And one side is where it transfer the material. So I hope that helps. Yes. One will be on the receiving side and one will be there on the exiting side. So cell theory was given by initially two scientists, Skelton and Schwann, right? They said that cells are the structure and the fundamental unit of life and all living organisms are made up of cells. Then Rudolph added an important postulate that cells arise from the pre-existing cells. Very good. Now moving ahead to the next one, which is lysosomes. I think, who was asking? I forgot the name. But lysosomes are also called as the suicide bags of the cell, right? They contain the hydrolytic enzymes or the digestive enzymes. So, if there is any waste material or if there is any pathogen which have entered, what these lysosomes will do? They will go and they will destroy it by releasing the hydrolytic enzymes or the digestive enzymes. Just in case the cell is not performing well, right? Cell activity is not well. What they will do, they will release the enzymes, right? And the cell will die. That's why we call them as the suicide bags of the cell. This is important, everyone. Common question asked, right? Yes, Anant was asking. I hope that, Anant, you got the answer. So it will actually help in the breakdown of the molecules, different foreign materials, and sometimes the worn out organelles also. For example, mitochondria is not working in the cell, right? So this lysosome will detect it by the signals that are moving, and of course it will go and destroy that mitochondria. Are we clear? Very good. Very good, Shweta. So everyone, are we clear? Quick thumbs up. Yes. Purna, you're asking that what does it modify? So what happens? We are getting the proteins and lipids, right? Now they can be in n number of shape. There will be a little requirements, right? Modify in sense that if it's properly structured, right? So they will do their modification. For example, let's suppose you go to a showroom, right? To get a dress. And you like the dress and you bought the dress. But you feel that the height of a dress is little bit low. So what, little bit high. So what you'll do? You'll go to the tailor. They'll modify here and there a little bit, right? That's what the lysosomes will do. They'll make sure it is properly modified so that it can easily get packed and go to the required place where it is basically required, be it the lipids or the proteins. Yes, contain membrane bound structure. Yes, it has one membrane. Very good. Nice. Now everyone, till now we have discussed about the animal cells, right? Now of course there is one important part which is the cytoplasm. Now cytoplasm is a jelly fluid, right? Jelly structure fluid which of course has all the organelles. A lot of reactions occurs, right? And all the cell organelles are freely suspended in the cytoplasm. Yes. Very good, Rashmi. Cis side, right? Shivanshu, Rashmi is answering. Cis matlab on the receiving side and trans matlab on the transfer side where towards the ER so that it can transfer. Now we'll be discussing about the plant cell. Now many of you asked about the plant cells, right? Now plant cells have the cell wall. That's the biggest difference. Then of course the second difference is the vacuole. Plant cells have big vacuole and vacuole plays a very, very important role in the storage, right? In both the plant cell and the animal cells, vacuoles are present, but in animal cells, the size of vacuole will be really very small. And of course, in plant cell, they provide the turgidity to the cell. It actually helps the plant cell to maintain its structure. That's the role of the vacuole. Then of course, only plant cells have the cell wall. Right? Animal cells have no cell wall, but plant cells have the cell wall. And cell wall is rigid, right? It is dead and of course it provides the protection to the plant cell. Right? Very good. Then comes the very important part you were asking about. The kitchen of the cell is called as a plastid. Specifically we will talk about chloroplast, right? Which has a green pigment called as a chlorophyll. And over here we will see the process of photosynthesis happening. It has a green pigment. Right? The other type of chloroplast is chromoplast, which gives beautiful color to the flowers and to the fruits. That's the chromoplast. The last one we have leucoplast. Now, leucoplast actually helps in the storage, right, of starch, oil, fats, etc. And it is usually found in the roots, which has no pigment, white pigment. Very good. Are we clear, right? We are clear up to here? Yes. Yes, chloroplast is also called a semi-autonomous organelle. Plastids also have their own DNA. The very important point. They also have their own DNA. Which part is of the cellulose? Cellulose is present in the cell wall. Yes, plastid is only present in the cell. Very good. Very, very good. Okay. Are we clear with this? So if we go back, we have discussed about the organelles. Yes. Now ribosomes, I'll say that, I'll write over here, right, ribosomes. Yes, ribosomes are very small structures, right, and they are present on ER, that too rough ER, and they help in the synthesis of protein, right? Prachi, because mitochondria and plastid both have their own DNA and they are capable of producing their own proteins. Good evening. Yes, I think many of you are here. How many enzymes? Now, Ashraf, I'm not sure you're talking about it, but enzymes act as the catalyst. They actually help in reactions to happen. And there are so many enzymes in our cells. Yes, are we clear up to here, everyone? Yes, quickly thumbs up. See, we are going really very slowly and we have already covered this. I don't know, I think by now you should have everything in your mind. Devdas, we will have them empty, but as of now we are learning. So first we'll learn and then of course we'll have the quiz. Stroma and vacuole, we don't have that. Stroma, you're talking about the stroma, that we of course, we have stromata, which is a different part, which is a different, it is there on the epidermis of the leaf, right? Basically it is there on the epidermis. It's present in the leaves, right? And of course stroma, that's there in the chloroplast. Now with that, we are on the final topic of cell division. Cell division is of two types, mitosis and meiosis. What will happen in mitosis? That the daughter cells will be the identical copies of their parents. Very good, Rashmi, but got your point. Right? So in mitosis, the parent cell and the daughter cells will have same number of chromosomes, right? Daughter cells will be the identical copy of parent cell, and that division is called as mitosis, equational division or equal division. The number of the chromosomes will not be getting half. For example, over here what we have? Diploid. Two sets are there, right? So it is that. Now in this mitosis, it occurs in the skin. It occurs in the stomach, the cells that we have over there, right? Very good. Very good, Shweta. Diploid to diploid, right? The chromosome number will stay intact. Very good. So if I write over here that you all are mentioning, diploid state, the number of chromosome will be in the pair. We represent it as 2n. Yes, and then the next type, of course, we have the meiosis, right? And meiosis is called as a reductional division. Now in meiosis, we know that the number of chromosomes will get half from the diploid, as you see over here, to the haploid, right? See the number of chromosomes being reduced from 46 to the 23. And in mitosis, we'll have two daughter cells formation, and over here we have 4. 1, 2, 3, and 4. 4 daughter cells are formed. Are we clear? At the breakdown of pyruvate to give CO2, water, energy takes place in mitochondria. But that's a class 10th question, not class 9th question. Are we clear? What are semi-autonomous organelles that get up? What an interesting name you have. So basically these are the organelles that are capable of making their own DNA. They have their own DNA, not capable of making their own DNA. They have their own DNA and they can make their own proteins. They're kind of independent in that way. Semi-autonomous, right? They are not fully independent, but they are semi. Very good. Ashraf, now you're asking any question. Yes? Very good. So are we clear everyone about the two types of cell division? In the CBSE, they have not gotten into the details of it. Right? In the cell division, if we say. Meiosis will occur in the gametes, in the production of sperm and the ovum. Right? Parth, diploid and haploid are the number of chromosomes. Basically, when we say na, haploid cells will have half the number of chromosomes. Over here, this is the haploid state. N. Diploid, it will have double the number of chromosomes. Yes, Monalisha, how plants cell divides, of course, we'll see this division happening, right? And they will have the mitosis. Do you think we will have the meiosis? What do you think? Think about it, everyone. Zygote means after the fertilization of sperm and ovum, we'll see the formation of zygote. Punya, I have been looking for this question. I don't have the answer for this. This is kind of not a logical question, right? I don't know. I think scientists just came up with this thing. Nice. Everyone, are we clear? Come on. I think we have covered up all the important points. Why the number of chromosomes reduced to half? Because of the cell division, right? What happens? The number of chromosomes, the chromosomes get mixed and there's a division happens. And then during the division process, only half the cell will receive only the half the number of chromosomes. And that's why over here we have 4 cells formation. So half of this will go. So if we have 46 chromosome, half will go in this and half will go in this. Very good, everyone. I think this is clear. Awesome. Now I want to see a very quick chain of smileys and thumbs up, everyone. After a lot of hard work, we all are here. Yes. Are we clear? Yes, very good. Very good, very good. If you have any doubts, please ask me. I'll just stay for here, we'll just, if you have any doubts, if you have missed anything, we can quickly discuss over here. Function of ATP. ATP is adenosine triphosphate. This actually gives the energy. It's an energy molecule, right? So whenever there is an energy requirement in the cell, in any of the reactions, ATP will go and help in the conduction of the reaction. Ma'am, they were 46, but how they become 2 cells of 4s? So that happens because of the cell division, bache. Yes, over here we have a cell. It has 46. During the division, what will happen? First division, 23 will happen. Right? That's how if we talk about it. Right? Now, of course, we can see this. Now we can see the number of chromosomes getting how? From 2n to n. Yes. Neelam, M phase is mitotic phase where we'll have the different phases, prophase, metaphase, anaphase, telophase. We will have menti for sure, but not today, but some other day. Yes, are we clear? Quickly, everyone. Happy faces, right? Awesome. Ronak, you can watch the video, bache. Again, it is not in the CBSE. If you want to learn more about cell division, we have done a detailed video on cell division. You can definitely watch it. Yes, very good. With this, everyone, we are done, and you know that all the teachers are talking about it. So, 7th of August is a very important date. So, stay tuned, right? You know that BYJU'S have got you covered. We are there for you. Here's a proof of the hard work. If you stay, if you stay with yourself, you study properly, and that result is really very, you can easily achieve it. And with us in the picture, we'll have amazing results. And with that, don't forget to hit the like button for the video. Share with your friends and subscribe, everyone. We really want to reach the 100K. So we really want your help with that. If you're new to the class, please subscribe to the channel. Please subscribe to the channel. Yes. Mitosis is a type of cell division where, of course, we'll see the equal division. Just remember, you have skin, right? Now, each and every time if you get a scar or a wound, what will happen? The skin will repair itself. Same type of cells will be coming here. So, in mitosis, equational division, number of chromosomes remain equal. Yes, Himani and Law, you have to wait. You can think about it, but it's a very interesting thing that will be happening. Sharanjit, welcome to the family. Subscribe. Thank you, Sumit. Thank you, Sumit, for subscribing to the channel. Everyone, please subscribe, right? And how many of you have liked the video? For your teacher. That means a lot. That means a lot. Now, I understand this. There are times when, what happened that you might feel that, ma'am, it's too much. There are few still some doubts. I would request you to watch the previous video that we have already done on the specific topics that will be helping you to learn better. You can watch them on 1.5x. You don't have to listen, right? Like you can just quickly make them on 1.5x and very quickly you'll be able to have through, you can actually listen all the session. Reductional division, Ronak means basically we call meiosis as the reductional division, which is nothing but the chromosomes will be getting to half and will have 4 cells at the end. You watch it at 2x, very good, yeah. That is also good. Nice. So can we close the class, everyone? I hope that you have enjoyed and I hope that this session have helped you a lot in clearing up your doubts and a very quick revision of the fundamental unit of life. Now, even after, I can see that, even after, like we have been learning this for a very long time, but I can still see there are students who have doubts. So I would request to all of those students, please read your NCERT, right? That will be helping you. Yes. Thank you, Ronak. Thank you, Priyanshu. Thank you, Manju. Very good, everyone. Awesome, everyone. We'll be meeting in the next class till that time. Take care of yourself. Lots of love, everyone. Lots of love. This is the way I think the new science, right? Lots of love and we'll meet soon. Bye-bye.